coming to the next stage of the fertilization process. We'll be talking about the embryonic development. In the previous video, we have talked about the insemination and the events which are taking place so as to facilitate the process of fertilization. Now, after the fertilization happens, following changes are found to take place in the zygote. Let us try and understand that once, if we draw the structure of the oviduct, ovary, and over here is the uterus part. Let me draw this over here. So once the ova, this is the ovary over here, and from the ova, after the ovulation process, that is let me mark it with the black over here, after the fertilization has taken place in the ampulla region. This is the junction of ampulla and isthmus. So over here, the fertilization has taken place and resulted into the formation of zygote. Now, once the zygote is formed, then it undergoes a division. That is what we call it as the first stage of embryonic development, which is known as cleavage. So as soon as the zygote is formed, it undergoes mitotic divisions, and these divisions are over here where it is holoblastic. The kind of division over here in which we see in the case of zygote are holoblastic because the amount of the yolk which is present is very, very less. So there is no problem relating to the division, and it results into the formation of two-cell structure. Now here you can also see that all the layers, that is the zona pellucida layer is there, the corona radiata is still there, the vitelline membrane is also there, and also the zona pellucida. So at this stage, we are saying the corona radiata, all the three layers are intact. So the zygote, as it is moving through the isthmus, it moves towards the uterus. After the division has started, the zygote will now move into the fallopian tube further, and there the division of the embryo, that is the zygote, newly formed zygote, will be taking place. Now, this cells which are being formed as a result of the division of the zygote, it results into the cells which are known as blastomere. Now, after the first division, which equally divides the cell, that is a holoblastic division, it later forms the 4-cell stage, that is the blastomere. The division will be like this, and it will result into the formation of blastomeres. So, in the case of cleavage, which is the first stage, as soon as it is moving, after the ovulation has taken place, these three are the ovulation part. So, after the ovulation has taken place, the fertilization happens to take place in the ampulla region. This is infundibulum. Infundibulum region. Now, here the fertilization has resulted, and soon after the fertilization, the zygote will undergo the mitotic division, and it will be a holoblastic division. And it starts as the zygote moving towards the isthmus and finally will land into the uterus. So it results into the cells which are known as blastomere. So as a result, over here also, the cells are continuously dividing, and all the layers, that is the 3 layers, are still intact over here. Now, after the blastomeres are formed, they will undergo rapid mitotic division, and as over here, the Moriola stage is next, where the number of blastomeres which are present within the ova, that is the fertilized ova, It is 8 to 16 blastomere stage, or sometimes the number of cells increase up to 32 also. And it is a highly compact structure where corona radiata disappears. So over here, this is the red one, which I have marked it as black over here. This red one, corona radiata disappears. But you can see over here in the morula, the zona pellucida still persists. And these are over here compactly arranged morula stage. So from here, this is the morula stage which is continuing till here. So isthmus region, we have the morula stage. Later, now this is the endometrial lining over here of the uterus. Now, this 8 to 16 cell stage, which is a compact cells which are compactly packed, and the corona radiata has disappeared with the zona pellucida still sustaining or still present over there. And these are very compactly packed. And when we observe this in the morula stage, we find that the cells are almost the same size, and it next leads to the formation of blastocyst. So the next stage after morula is the blastocyst, where the rearrangement of the cells of the morula takes place. Now here the blastomeres will be rearranging themselves, and here the outer, that is the layer, zona pellucida, now disappears in the next stage. So we have seen that from the zygote stage to the stage of the morula, all the three layers are present before the morula formation. Corona radiata disappears at the morula stage, and zona pellucida disappears at the blastocyst stage. So here the blastomeres are rearranged, and the zona pellucida, as you can see, is not present. Instead, all the blastomeres will arrange themselves in such a way that small blastomeres will be present at the border. These are the blastomeres which are present at the border, and the bigger cells, which are marked as blue, will form the inner side. So they will be arranged in the inside of the, and these are slightly bigger than the cells which are present outside. So these cells are then classified as, the outer layer is known as the trophoblast, whereas the inner layer is known as, or the inner mass of cell is known as inner cell mass. The trophoblast therefore provides nutrition and protection, whereas the inner cell mass will develop into the embryo. So this complete structure is known as blastocyst, and it is characterized by the presence of blastocele, that is a fluid filled cavity due to the secretions of the trophoblast and the inner cell mass. So blastocyst is a next stage where the zona pellucida is absent. The inner cell mass formation is there. The trophoblast layer is also formed, and this trophoblast layer forms the outside of the cell, whereas the inner cell mass will later on develop as the embryo. And there it has, the blastocyst has a fluid filled cavity. It is all fluid over here, and this fluid filled cavity takes care of the embryo. So we see that zygote moves from single cell to two cell stage to the 4 cell stage, that is the blastomeres which are being formed, to a compact structure which is 8 to 16 or maybe 32 cell structure, which is known as moriola. And this moriola, here the rearrangement of cells of blastomeres, we call it. rearrangement of blastomeres, and it results into the structure which is known as blastocyst. So this blastocyst is the next stage where the development will be followed. Now next is, now over here, this happens around the third day. Now, the implantation, it is a very important phenomena which takes place, and that is a process in which the blastocyst becomes embedded in the uterine wall. So you can see over here, the blastocyst which is now present over here this is the uterine thinning lining, that is the endometrium. Now this endometrium at the time of implantation is very, very thick and richly supplied with blood capillaries. So the next stage is the implantation, where the blastocyst, now this is the blastocyst which will be embedded in the uterine wall, and it occurs after 7 days of fertilization. So this is the third day, and at the 7th day of fertilization, this blastocyst will be embedded or will be taken up or will be implanted in the uterine wall, where the further development of the embryo will take place. Now, this involves the rapid division of trophoblast and release of enzymes by that. So, what is happening actually over here? This is the lining of the uterine wall, that is endometrium. Now this line or this lining needs to be corroded by the secretions from the trophoblast, and this as a result will allow the blastocyst to fit into the depression which has been created due to the depletion of the endometrial lining. Now after that, the villi, that is the chorionic villi, appear from the trophoblast and which are surrounded by the uterine wall, and as a result that leads to final implantation, or we call it as pregnancy. So over here during the embryonic development, we find that the zygote has started from a journey of single cell. Right from here, this period is marked as ovulation over here. This is the ovary. So the fimbriae will collect the ova which is being released, and after the fertilization has happened, the division of then after the fertilization, the zygote that undergoes division and will form the blastocyst, and this blastocyst will be finally embedded or implanted within the uterine wall for further development, and that is what is known as pregnancy. In the next video, we'll talk about how the implantation has taken place and the chorionic villi and later development of the embryo. Thank you.